where there are some geometrical figures, and please don't use the chat for your question. Now I can leave the floor to Nicolas Science,
Yes.
that will introduce on behalf of E3S board. Sorry, Nicolas.
Thank you very much for your introduction. On behalf of the E3S board, it's our pleasure to welcome you to the last webinar of E3S 10th anniversary series event. So the next event will be the E3S Annual Symposium and General Assembly, which will occur on Monday and Tuesday, 9th-10th of May 2022 at Reading in the UK. But today, the webinar is called Take a Seat at the Table of the Future, and it is organized by the Next Generation Group. The group was formed in 2015 by Martha Skinner and a small committee. Then I took the lead. I was the chair for several years, and now the group leader is Karina Gonzalez-Estanol. And the group and the mission of the group is to allow students and early career researchers to gather and share knowledge with fellow scholars. They are organizing multiple events such as webinars, gathering meetings, and so on, to strengthen the network among students in Europe. So now it's time to start, and I leave the floor to Karina and Maddalena for the webinar. Thank you.
Yes, thank you, Nicolas, for the nice words and the introduction. It's great to see a lot of people joining our seminar today, this afternoon. Indeed, this is the last webinar as part of the 10-year anniversary of the society. So yeah, let's take a seat at the table of the future. As Nicolas already said, my name is Karina Gonzalez, and I'm here with Maddalena Libardi. And together we are part of the new committee of the Next Generation Group. Yeah, so then basically one of the, as Nicola already said, one of the objectives of the group is to create network among young researchers. And I'm very happy to say that now we count with more than 80 members from more than 11 European countries. And we hope to continue growing this very nice scientific community. Because in fact, yes, we are creating a network across Europe, but also we're providing a platform where we can communicate and exchange our ideas with each other. But more important, we're making visible our research projects and our work, not only at different conferences, but also at events like this one.
Yeah, and as Karina just said, one of the results of the communication between young researchers is the webinar that we organized today. Today we have the opportunity to have 6 amazing researchers that are going to present their work that is done either in the academic or industrial work. So we are going to first hear about 3 speakers, which are Hannah Ford, Lauren McGuinness, and Hannah Liani. And we're going to have a discussion table where you can address the question to these first 3 speakers. We're going to then have the 3 last speakers for this webinar, which are Maria Isabel Salazar Cobo, Luca Balog, and Leonardo Menghi. And we also then will have a second round of discussion table when you can address these questions to the speakers. As Ilona already mentioned, some webinar housekeeping rule, please make sure to be muted and use the Q&A feature for asking your question that will be addressed in the discussion table. Make sure please to have type down the name of the speakers and the question that you would like to ask. So now, without further ado, I'm going to introduce the first speakers of today, which is Hannah Ford. Hannah is a PhD candidate at the University of Nottingham, and she's going to tell us more about her study, which is entitled Exploring Meat-Eating Consumer Willingness and Motivation to Reduce Meat Intake and Accept a Protein Alternative between Australia, China, and UK. Please, Hannah, you can share your screen and start your presentation.
Lovely. Thank you very much for the introduction. So I'll just get my presentation up. And hopefully you can all see that. If not, yeah, let me know. Good. Okay, yeah, thank you for the intro, and it's lovely to be invited to present some work, which I'm excited to do so. So without further ado, for those of you that don't know me, my name's Hannah Ford. I'm a third-year PhD student on a joint programme between the University of Nottingham and the University of Adelaide. And my PhD overall is looking at understanding consumer behaviour towards sustainable food and beverages. So it's a very broad topic, but a very hot topic area of research, I'd say. When I started out with my literature review, I realised quite soon that meat consumption was a very prevalent area of research, and to some degree underpins the success of sustainable diet. So it was something I wanted to explore a bit further, which in this presentation I'm going to give you a snippet of some of the research I've been looking at so far. So you might be asking, well, why do we need to reduce our meat consumption? In fact, this is often a bit of a controversial topic for many because it does play such a significant role in our social and cultural lifestyles. And for many of us, it tastes really good. So it's hard to give up. But unfortunately for meat, it's one of the biggest contributors to food-related greenhouse gas emissions, yet consumption continues to rise at a very unsustainable rate. And it's a bit of a sobering thought, but if we continue the way we are, we will not meet the UN Sustainable Development Goals by 2050, which aims to keep global warming well below 2 degrees, aiming for 1.5, which of course will have disastrous knock-on effects. So when I looked a bit closer at the countries I was studying, so Australia, the UK and China, it's quite interesting to see the different meat consumption patterns. So in the UK and Australia, although they've been quite consistent, steady, over the past few decades, they've been relatively unsustainable in their rates. And Australia, I believe, is still in the top 3 biggest consumers of meat. And then, as you can see, the red line of China, how fast and rapidly their meat consumption is increasing, and it's predicted to continue with this positive trajectory. So it is a bit alarming, and I suppose more needs to be done to change behaviour in some way. So my research questions hope in some way to address this issue. And you're probably aware in 2019, the infamous Eat Lancet Commission report came out, and it was the first report to highlight a global transition shifting consumption behaviours, and it recommended more than a 50% reduction in red meat. Slightly controversial, obviously, depending on where you are in the world. And then in recent months, a good example is the International Panel on Climate Change, their report, which highlighted how important lifestyle changes are to mitigate this greenhouse gas emission effect. So for the first time, we're starting to see meat reduction, not just for health reasons, but now for planetary environmental health. Therefore, my questions are quite straightforward. They're in my intro. So I wanted to understand how willing consumers are to reduce meat, what their motives are, and then repeat that for 3 protein alternatives. And underpinning all of that, I really wanted to understand to what extent do these factors differ across countries. So in terms of my materials and methods to address this research question, I did an online consumer survey, which is everyone's friend during a pandemic, and I looked specifically at meat eaters. So I distributed the surveys across the UK, Australia, and then an Emirates student, Yuchen Zhang, collected all of the data for the Chinese sample sets. I have to thank him, and I specifically looked at Shanghai, just to mention that. It was part of a larger survey called the Sustainable Dietary Study, so I collected an awful lot of information, also relating to meat attachment, personality traits, consumers' perceptions around the environmental impact of food and sustainable diet. So a lot of data, but in this talk, I'm just going to be focusing on that willingness to reduce and the motivations behind that. I took the 7 motives that were deemed most important from 2 validated scales, the Food Choice Questionnaire and the Ethical Concern Subscale, and I repeated these questions for 3 protein alternatives, which were meat substitutes, edible insects, and cultured meat. So they're deemed pretty different to each other, so I thought I'd get some interesting results in terms of willingness and motives. So on to the results, and it's very interesting finding that Chinese and UK consumers were significantly more willing to reduce their meat consumption compared to Australian consumers. Only 36% were willing. And then in terms of the motives, well, this graph is looking at strengths of motivation. So it was on a 7-point Likert scale, 1 being extremely unimportant to 7 extremely important, and 4 was the neutral score. But the first thing I'd say to highlight is the Australians in the green line, their overall motivation was significantly lower than the UK and China. So as expected with that lower willingness. Worth highlighting is food safety was the most important factor in China, and this was significantly higher compared to the UK and Australian consumers. Whereas in the UK, the environmental benefits were the most important factor, again, significantly higher than China and Australia. I thought it was also interesting to look at animal welfare and the big gap between the UK, which found this significantly more important compared to the other two countries. In terms of the results for willingness to adopt protein alternatives, and this bar chart simply shows that willingness in the UK and China was significantly higher for meat substitutes and cultured meat compared to Australia, whereas edible insects, there was a bit of a change in pattern there. It was the UK and Australia, perhaps not what we expected, was significantly more willing to adopt compared to China. And the percentage for preference, so the UK and China, their most preferred alternatives was the meat substitutes, although followed closely by cultured meat. Whereas interestingly, in Australia, they preferred edible insects, so a bit of a change in the order of preference. And then the last result is looking at the motivations to adopt protein alternatives. And I appreciate these charts are probably very small for you, so you won't be able to see the post-hoc groupings, but I just wanted to show overall the trends. So each graph represents each country, and I think the key point really is Australia again. In the red line is their meat substitute overall motivation, which was significantly lower than the other protein alternatives and compared to China and the UK. It's also worth saying again about food safety, higher in China, but this time we started to see a difference between the protein alternatives, so it was significantly more important for the case of edible insects and meat substitutes compared to cultured meat. And then sensory appeal in the UK, if you follow it across, you can sort of see that it's higher for some protein alternatives compared to the other countries, especially for those edible insects and cultured meat. Not so much for meat substitutes, maybe because we've tasted the products before. So, and then the last point, sorry, is welfare. I thought that was quite an interesting one to see the gaps between some of the protein alternatives. So the UK, as expected, the welfare potentially for the insects is less important compared to the others. So that was a very brief overview of some of my results, but I really do hope that they show how cultural factors can inherently influence willingness and motivation to reduce meat and adopt protein alternatives. But they also highlight the need for country-specific meat reduction strategies. I don't think one solution will suit every country, and that's probably best highlighted by the fact that Australians had that higher preference for the edible insects. So potentially it's worth asking, is it better to develop those kind of products than meat substitutes? It's worth further thinking. But in terms of further analysis, well, I've got a lot of data to be keeping me going because I'm also looking at other important factors such as meat attachment, environmental perceptions, and personality traits, and whether they are good factors to predict intention to reduce meat. So all that's left to say is what will be at the table of the future. How I interpreted it was perhaps a bit dark, but like I said, will it depend on the choices we have left, which is a very prevalent question. I think that's worth asking. But thank you very much for listening. If you have any questions, feel free to email me or add me on LinkedIn. Otherwise, I look forward to answering the questions you have at the end of the session. Thank you.
Thank you, Hannah. That was a very fascinating and indeed a very hot topic. And I'm sure you will receive a lot of questions that we can address in the discussion table.
Thank you.
But now let's move on to our second speaker, Lauren McGuinness. She's a PhD candidate in sensory science at the University of College Dublin. And she will tell us about the consumer's perception of grass-fed dairy products. So the floor is yours. Lauren, please share your screen.
Hi everyone, I'll just share my screen now. Sorry, can you see my presentation?
We see your screen shared. If you click on PowerPoint, yeah, I think if you start a presentation from there, we will see it.
Yeah. Okay. So you can see that now, yeah?
Yeah.
Okay, perfect. Sorry. So My name is Lauren and I'm a PhD student working with Food for Health Ireland in University College Dublin, and my work examines the effect of different feed types on the sensory properties and consumer perceptions of dairy products. The study that will be presented today investigated the effect of front of pack claims on consumer perceptions of cheddar cheese in both an Irish and US cohort. So why are we interested in grass-fed dairy? So in recent times, there's been an increase in plant-based sustainable diets with a move away from dairy products, but not all dairy is the same, and some may be more acceptable to consumers than others. Grass-fed dairy is perceived as being healthier, more natural, more sustainable, and more animal welfare friendly. Recent studies have shown that consumers are willing to pay more for grass-fed milk, and grass-fed milk is considered a higher value product that is also meeting needs for more sustainable dairy production. So to put context to grass-fed labelling, Board BIA, the Irish Food Board, have recently introduced a grass-fed standard in Ireland where an individual herd must be fed a minimum of 90% grass or grass forage on a fresh weight basis. Cows must spend an average of 240 days per year at pasture, and milk pooled for processing from these farms must achieve a minimum grass-fed figure of 95%. A similar strategy is used in the US, where organic farming requires cows to spend a minimum of 120 days per year at pasture for a dairy product to be labelled as pasture-fed or pasture-raised. So we wanted to understand if and how this grass-fed claim impacts consumer perceptions and or potential portion selections when it's added to a front-of-pack label. So grass feeding is typical of temperate climates, for example, in Ireland and New Zealand, while indoor feeding systems using total mixed ration diets is more common in other climates, including the US. Milk derived from a grass-fed diet has a higher fat and protein content with improved nutrition content, higher concentrations of acidic acid, conjugated linoleic acid, beta-carotene, and alpha-linoleic than TMR-derived milk. But these differences in feed type also lead to differences in milk composition, which influences the sensory properties of the milk and dairy products. Grass-fed dairy products typically have grassy, cowy, mothball, and barny flavours, with increased yellow colour and spreadability, whereas TMR-derived dairy products typically have malty and sweet aromatic flavours, are harder, and are less yellow in colour. There is some evidence that consumers differ in their perceptions of grass-fed dairy products depending on the feed type common in their geographical region, and that these differences are likely influenced by familiarity. So our study aimed to examine the impact of a lighter fat and or a grass-fed claim on the expected characteristics of cheddar cheese, the portion size selected for cheddar cheese, and if these expected characteristics differed between Irish and US consumers. So participants from Ireland and the US completed an anonymous online survey where they were shown mock cheddar cheese packs with different labels, as shown on the screen here. So they were either shown the full fat label with the grass-fed logo, or they were shown the lighter fat label and the lighter fat label with the grass-fed logo. Based on each label, participants were then asked to rate their expected liking, flavour, intensity, aftertaste, perceived healthiness, and perceived naturalness of the product. And participants then selected a portion of each cheese they would likely consume on a slice of toast for lunch from 100 portion size images ranging from 1.2 grams to 120 grams. And this was repeated for each cheese label. So what we found was that grass-fed claim did not influence expected sensory characteristics, but increased the healthiness and naturalness perceptions in both cohorts. The lighter fat claim decreased expected liking and flavour intensity in both cohorts, with US participants expecting a lower liking and flavour intensity from lighter fat cheese compared to Irish participants. The lighter claim increased perceived healthiness of the cheese in the Irish cohort, but US participants perceived the lighter fat products as being less healthy than regular fat products. And the front of pack claim had little impact on the portion selection, with the exception being with Irish consumers who chose a higher portion for the lighter fat claim in comparison to the regular fat grass-fed cheese. This was not seen in the US cohort. So we concluded that a grass-fed claim may enhance consumers' perceptions of healthiness and naturalness and may offset expectations associated with lighter cheese. But regarding the practical implications for industry, it's important to note that consumers have a positive perception of grass-fed cheddar cheese without compromising sensory expectations. On the other hand, consumers may increase portion sizes for lighter labeled products, reducing the effectiveness of these foods for weight control strategies, which has implications for public health messaging. So moving forward, we will be extending this research to other populations, including in a Chinese cohort. And we will also be investigating the differences in sensory characteristics of grass-fed versus non-grass-fed dairy products using trained and consumer panels. So before I finish, I would just like to thank Food for Health Ireland for funding this work. Thank you to E3S for allowing me to present to you today. And thanks to my supervisors, Dr. Emma Feeney and Professor Dolores O'Riordan, as well as the research team who worked on this project. Yeah, so thanks everyone for listening. My email's on the screen there if you want to get in contact. LinkedIn as well under Laura McGuinness.
Thank you so much, Lauren. It was indeed a really incredible research, and I really look forward to see how the result will go when you extend to other populations. Now we're going to move to our last speaker for this session. So our next speaker is Hannah Hyliani, and she's from the University of Bremen. And she's going to present us her study on the changes in taste perception and preference due to growth and maturation during childhood. Please, Hannah, the floor is yours. I think you need to turn on your mic, but we can see your presentation.
Okay, thank you. So now I have to find the presentation again. So I think now you can see it.
Yes, we can see it.
Okay, thank you. Thank you for your introduction, and I'm really happy to be here and to present to you my postdoc project, which I actually just started. So I'm going to tell you what I'm planning to do. So first I will tell you about the background, which led to my research question, and I will show you the methods I'm planning to use to answer the research question. And then I will show you the result, which I'm aiming to achieve. So first, I can say that during my PhD, the most striking result that I got was that almost every dimension of sensory perception, like sensitivity and preference, was associated to age. So this led to the question if the process of growth and maturation during childhood has an influence on sensory perception and preference in order to consume maybe a larger amount of energy during growth spurts that occur during childhood. So yeah, this was my research question. Like I just said, if the specific markers that are elevated during growth can alter the taste perception and preference. So I am lucky and I can analyze the data of two really large cohort studies that were conducted in Europe in 8 European countries and that investigated children between 2 years and 17 years and even measured the same children several times. So in a subsample of these two big cohort studies, also sensory tests were conducted.
Hannah, sorry to interrupt you, and I think we are not seeing your presentation correctly because we are still in the first slide, so I'm not sure if there is, it's not the correct sharing.
Okay, so maybe I can stop it and start it again. I don't know why, but I can't. I can't select the presentation. So I will just close it and open it again. I don't know. I can't. I can't. I can't select the presentation.
We can try to share your presentation if you want. We have a backup here.
Yeah. So do you see anything or not?
Now we see. And if you try to change the slide.
Yeah.
Okay, now I think it works. Okay, perfect.
Okay. So these were the results from my PhD, which I just mentioned, that I found that every dimension of sensory perception was associated to age. And I was asking myself whether this is associated with growth and maturation during childhood. And this led to the research question of my postdoc project. Yeah, and I just started, and I'm aiming to analyze data from these 2 large cohort studies that were conducted in 8 European countries. And just to give you some numbers, so the specific numbers of the analyses, they depend on every analysis, but this is just to show you that in the first cohort study, about 2,000 children participated in the sensory tests. And in the follow-up study, the same children plus their siblings and also their parents participated in the whole study and also in the sensory tests. So these were the first tests that we conducted, a taste threshold test to measure taste sensitivity, and then also taste preference tests. As you can see, we conducted the tests like a game with the game board to engage the children. And we also conducted the preference tests with real foods and drinks, to have a more real life character of the results. And then we also applied a taste preference questionnaire where we asked the participants for their preference of the taste of different foods, drinks, and dishes. And from the results, we calculated age and sex-specific taste preference scores for sweet, salty, fatty, and bitter. Further, we also collected data on diet. So we applied a food frequency questionnaire to all children and also to the other participants, like parents. Then we collected saliva, urine, and blood. And also in a subsample, even smaller subsample, we conducted fMRI scans. Yeah, and then in my postdoc project, I also plan to involve a group of children actively throughout the whole project, because the literature says that involving the target group can enhance your results, because you can always discuss your research question, your methods, and your results with the target group. And the children, they can tell whether you are asking the relevant questions or which methods might fit more to investigate children. So with the data that we collected, I plan to do the following analyses. first, I plan to investigate the association between markers of appetite and markers of maturation, like, for example, tender stages, with taste, perception, and preference. Then secondly, I aim to analyze the association between markers of bone metabolism as a surrogate for growth with taste, perception, and preference. And then also the fMRI data. I aim to investigate in association to taste. And I also plan to, maybe discuss with the target group or investigate also with the data different sensory methods to measure taste in children. So which methods suit best to the target group. And then I also plan to organize a symposium, which might also be interesting for you. And I will be very happy to discuss the whole project also during the conduct with those who are interested. And I plan to organize the symposium maybe in 2 years from now, to discuss the results and also other current research results in the field of children. And all of this, so the results of the analyses and the symposium, and also the results of the active involvement with the children, will go into a new research application, which I aim to submit at the end of my project, which also should be answering the research question maybe even more. So maybe I need to collect new data then to answer the research question fully. Because I think it's really difficult to investigate growth and the different phases of growth. I think we have to plan a longitudinal study in association with taste perception to really answer the research question that I've got. So thank you for your interest, and I'm happy to discuss with you about this topic.
Thank you, Hannah. That was a very clear, a great talk, and it looks like a very ambitious project, and I'm very curious to see your results and to discuss them further. We will now continue with the first discussion table. We have received some questions on the chat, and we also have some questions for you. So let's just start. This question is for Hannah Ford. Hannah, did you look if there are any disparities between different education level groups, so say in high school, university, or master degree?
Yes, so we did collect education information, and it was a bit of a skew, I must say, towards the Chinese being more educated than the Australia and the UK. But because it wasn't a main focus, I haven't delved into it properly, so I can't really say if the willingness to reduce changed in any way. It was more about gender and age, where we did see differences, with the men being more willing, or less willing, sorry, to reduce than women, which is kind of what we expected. Yeah, so it doesn't really answer it, but it sort of does.
Okay, thank you. Another question, how did you define the flexitarian group?
Yes, so the flexitarian, all the dietary options had definitions, and the flexitarian was meat eaters. I'm trying to remember now. So it was different to omnivores. It was sort of following a vegetarian diet, but occasionally eating meat once or twice a week, from what I recall. Yeah.
Yeah, that's interesting, because always when we talk about flexitarian, indeed, somebody thinks that their own perception of flexitarian, even maybe eat vegetarian once a week. So that's indeed, it would be interesting to standardize that terms. Thank you so much. We have some questions also for Lauren. Will your online survey research be verified with test trials?
Yeah, so we're in the middle of training a sensory panel at the moment, so they will carry out descriptive analysis on 3 different types of dairy products, and then we will also be running consumer trials for the liking, the aftertaste, flavor intensity as well.
Okay. Do you have any hypothesis on the result you will obtain with a Chinese subpopulation?
Yeah, so our survey that we're developing at the moment for the Chinese population is actually more broad than what we did for Ireland and the US. So we have a few extra dairy products put in there. And for us, I guess the main question we want to know is what dairy products are the Chinese population eating? And then we will also mirror that with the research that we've already done and look at portion sizes in relation to cheese and the other dairy products that we'll be presenting as well.
Thank you.
Thank you. Now we continue with our last speaker. A question for Hannah. I find it a super approach that you aim to involve children. Can you tell a bit more about how you recruited or aim to recruit and discuss the matters with them? Definitely a challenge, for sure.
Yeah, so thank you for this question. Well, when I wrote the design and application for the project, that was before the pandemic started. So yeah, I think before it was a little bit easier, but now actually I'm thinking about either I'm a little bit I have to rethink my design of this, but I think one possibility is to look for groups that are already established and try to meet them, and then I aim to conduct also sensory tests with them to see how they like it and which will work and which won't work. And then I'm also very interested to talk about healthy diet to them, because I think many children, they already know what's healthy and what's not healthy, but they still prefer the sweets and the snacks. And I really want to know why it is like that. I mean, why they, which strategies would work with children to convince them to eat more healthy. So yeah, it's actually both to try the sensory tests and also maybe a little bit sensory training to make them more sensible or sensitive to the tastes and everything, and then also to talk about healthy diet and how they can be, or how they can adapt to a healthy diet.
Thank you. And another question, have you thought about working with children with autism, for example? Because, for example, it's known that they have some difficulties with different textures, for example, because they're oversensitive in their tongue, right? So have you thought about working with this this group, or are you looking specifically in healthy children?
Which group? I didn't understand which kind.
Group, children with autism.
No, so far I didn't think of this. So do you think that's that would be interesting to?
Yes, because I know that when they are children and they are not diagnosed, they are oversensitive, or they could be oversensitive in their tongue, so they cannot handle certain textures or something. So it may be interesting to take a look into them. I don't know.
Yeah.
No, indeed. Well, maybe.
Yeah. Okay. We have time for more questions. Yes. How many children do you plan to involve in this in your future work?
So do you mean in the active involvement?
Yeah, in the active involvement.
Yeah. So my original plan was to establish a group, like not so many, just 10 children maybe, and then discuss with them throughout the whole project. So every step of the project, I aim to discuss with them. So if they think that's a good idea to do this and this and this analysis, or if they have other ideas or think that maybe different questions would be more important to children, or yeah. So yeah, I thought to recruit like a like a research group of children that will be there throughout the whole project.
Well, that's definitely a challenge, but sounds great. Good luck with that. So if you have any more questions or you would like to address them privately with our speakers, then you can feel free to always send them an email or a comment or write it in the chat.
But yeah, now I think we can move on with the second round of speakers. So the first person for this session is Maria Isabel Salazar Cobo from the Wageningen University, which is going to tell us more about the dynamic changes in hedonic and emotional responses to food varying in portion size. Maria, the floor is yours.
Good afternoon. Thank you for your introduction. I will share my screen. Am I sharing it now?
Yes, we can see it now.
We can see it.
Yes?
Yes.
Okay, thank you. Does portion size matter? Hedonic and emotional responses to food varying in portion size. Do you think that in terms of our indulgent foods, the bigger, the happier? Well, on one hand, it is acknowledged that liking ratings alone do not predict food choice accurately, and that assessing consumers' emotional associations with food adds power to the prediction, to the food choice prediction model. Also, it is well known that portion sizes are getting bigger and bigger. And this leads to overconsumption and adverse health consequences. Also, there is a research found that adults tend to prefer larger food portions over smaller portions. Also, they eat more, they consume more when they are served in larger portions. However, the expected enjoyment of larger food portions do not always match with the actual food enjoyment. This is why we designed a study to investigate the dynamic changes in hedonic and emotional responses to indulgent foods, ice cream and pizza, varying in portion sizes. We hypothesized that regular portion sizes would evoke more positive hedonic and emotional responses compared to either the small or the larger portion sizes. So the portion sizes that we use were for ice cream, it was the mini magnum for the small portion. For the regular was the regular ice cream, the ice cream magnum, and for the large portion was a combination of the small and the regular ice cream. In the case of pizza, for the small portion was 2 slices of about 37 grams of margherita pizza. The regular portion was 3 slices, and the large portion was a whole pizza of about 296 grams. In this study, we focused our attention on the first and last section of consumption because we expected that the dynamics within the emotional processing will be more explicit in these sections. In this within subjects study, 58 participants randomly consumed a small, regular, or large portion of ice cream and pizza during 6 individual test sessions. The test session consisted in 3 stages: before, during, and after consumption. Before consumption, we measured the state of hunger and arousal. Then participants received their food, ice cream or pizza, to perform the TDE and video record during food consumption. After consumption, we measure liking, hunger, overall satisfaction, and again, arousal to evaluate the change. For the temporal dominance of emotion and facial recognition, we ask participants to put the food on their mouths and select the emotion that they consider, that they perceived as dominant. They click on the start button on their screen and start selecting the emotions that they perceived as dominant. The selected emotion remain as dominant from the moment that they selected until participants select a new emotion. When participants reach the first section of ice cream or pizza, was the first line, they click on stop and freely eat their product until they reach the second line for the last section of consumption and repeat the process that I just mentioned. To analyze the data, we use mixed model analysis to evaluate the effect of portion size, section of consumption, and the interaction of both on the 6 emotions, arousal and balance. Also, we use the TDE band plot to evaluate and visualize the sequence of the dominant emotions for ice cream and pizza, the first and last section of each product. As we may see, the small and regular portion sizes for both products were mainly dominated by positive emotions, such as relax, happy, and peace. However, the large sections, especially the last section of consumption in both products, we may see negative emotions such as boredom, guilt, and disgust. Our main findings confirm that large food portions were liked the least for both products, for ice cream and pizza. And as we saw, the small and regular portions were dominated by happy, relax, and peace, whereas the last section of the large portion was dominated by negative emotions such as disgust, guilt, and boredom. These results indicate that the change or the variation of the emotional profile is not only between portion sizes, but within each portion size, as we saw between the first and last section of consumption. So better emotional experience for small and regular portions than for a large portion of these indulgent, high energy dense foods. But the question is, are these findings transferable to low energy dense foods as fruit and vegetable? So we designed the same study with low energy dense food, varying in portion sizes and section of consumption. In this case, we use a mix of fresh fruit salad containing pineapple, strawberries, and blueberries. The small portion was 60 grams, the regular one 150, and the large one 250 grams, which is 50 grams above the recommended intake for fruit. For the everyday, the fruit was clean, chopped, mixed, and served in these clear containers. And we draw 2 lines to identify, again, the first and the last section of consumption. We replicate the methodology with 45 participants, and we found similar patterns for the first and last section of consumptions of the small and the regular portion sizes. And the 2 smaller sizes, the small and the regular, were mainly dominated by positive emotions, whereas the large portion was characterized by feelings of boredom, especially in the last section of consumption. The consumption of the fresh fruit salad was about, in the large portion, was 120 kilocalories approximately, and it led to feelings of boredom compared to our previous study with ice cream, which was more than 300 calories, or pizza, more than 700 calories. This implies that with less calories, with this type of product, low energy dense products, with less calories, we had a faster onset of sensory specific satiety as compared to the more indulgent foods. So to promote healthier eating behaviors, one way is to offer smaller portions of these indulgent foods and still provide emotional rewarding experience and pleasure, as we saw with ice cream and pizza. Additionally, to increase fruit consumption and meet the nutritional guidelines for fruit, it might be better to serve smaller portions of fresh fruit, smaller portions several times across the day, instead of serving just one large portion in one single occasion, leading to boredom. To conclude, people enjoy small portion sizes of high energy dense food about as much as large portions, as we saw. And fruit is a highly liked food, but it gets bored when eating in larger food portions. I want to say thank you to my supervisors, Dr. Heri Yaher, Lisbeth Sangstra, and Kees de Graaf, who helped me in this project, and thank you for your attention.
We're back. Sorry for this.
Yeah, sorry, but I had some problems with the microphone. Thank you, Maria, for a great presentation. I remember I was part of this study, and so very interesting results that you obtained, and I'm sure we're going to have some interesting questions at the discussion. I will move on to our next speaker, Luca Balog. She just finished an internship at iQuestion, and her talk will be focused on implicit measurements using eye tracking glasses. So please, Luca, if you can share your screen and start your presentation.
Yes, okay. Okay, can you see it now?
It's loading. Yes, we see.
Okay, great, thank you. Okay, so hi everyone, my name is Luca Balog. I currently am a student at the University of Amsterdam on a Brain and Cognition Master track. And during my 3-month internship at iQuestion, I did a pilot study to validate Kexu's eye tracking glasses called OpenEye. Now, eye tracking has many uses in sensory research. It can provide us visual representation. It can help us determine the most looked at object, and we can also get information about cognitive processing. Now, for visual representation, we would mostly use a heat map to determine the most looked at object. We would look at the frequency and the length of the gaze. And finally, for cognitive processing, we would take into account things such as blinking rate and pupil size change. Now, in this video, you can see a recording of the eye tracking. On the upper left corner, you can see the eye that is being tracked, and you can see the markers signed in, and the little cross is the movement of the eye. Now, there are many benefits and disadvantages as well to eye tracking. The biggest benefit is that it is largely unbiased. It's very hard to influence consciously. It is very universal, and it provides us a clear indication of attention. However, it is still a very expensive equipment and is still under development. And also it has a limited scope in what it can tell us. Now, the equipment being tested here, OpenEye, has 2 different cameras. There is a 180 degrees camera that records the environment. This is a wide lens camera, and it was applied to record the whole field of view that a normal camera would not necessarily be able to do, as those are usually around 70 degrees. The other camera is a high-speed global shutter camera. This is the one that records the eye. Currently, there are plans being made to implement an infrared lighting here as well, because that would stabilize the accuracy even during low lighting conditions. And all of this is placed on a 3D printed base. Now, this was the structure that was used on this research. However, this is not the final version of the OpenEye. This is what it would look like, hopefully soon. And it is still a very lightweight equipment as not to restrict the movement of the head. Now, the goals of this research was, first of all, to test OpenEye. Second, to determine its compatibility with iQuestion's goals, and to provide feedback, and finally, to provide a base for future research. The research itself included 16 participants and was conducted in a supermarket. Participants were handed a shopping list made of 4 tasks, each of which were based on a different hypothesis. These were to pick something from 100% vegan shelf, to choose an eco-friendly laundry detergent, to choose a healthy cereal, and to choose the best quality pandan rice. Now, these tasks were shuffled for each participant in order to avoid bias. In the first 2 tasks, the fixation value was taken into consideration, while for the last 2, the in-view value. Post-processing consisted of labeling the markers of interest, training the AI, and finally exporting the values. Labeling happened with this tool that you can see here. There are the points. I'm not sure how visible they are, but those were the points that we could use to mark the different products that we wanted to measure. And this is how it looked like after the AI has already recognized them. So you can see the number of the point, and you can see the number of the value in seconds. Now, in terms of the export, the data analysis was done with an Excel file, but a graph was also presented. Here you can see that the blue is the in-view value and the green is the in-fixation value. Now, in the research itself, the 4 hypothesis was that eye-level signs will be fixated on more than the floor stickers when looking for the vegan shelf, that laundry detergent with simple bold lettering will be fixated on more than the green packaged ones when looking for an eco-friendly laundry detergent, that neutrally packaged cereals will be in view longer than colorful ones when looking for a healthy breakfast cereal, and finally, that the rice on the highest shelf will be in view the longest when looking for the highest quality pandan rice. Now, there were no significant results in either of these, probably due to the low sample size, but there were, in some cases, already indicative differences in the mean values. So the last 2 were already pretty well supported by previous research, so I will not be discussing those, but the first 2 were relatively new areas of research. Now, with the first task, the floor sticker was in view the most, which actually was contrary to what we expected. So these were measured first: the green value, the green sign, the white sign, and the floor stickers. Our hypothesis was based on the fact that eye level and a little bit higher than eye level areas are usually the longest in view for eye tracking studies. However, with the floor stickers, especially during and after COVID, as this was conducted when there were still regulations going on, the floor stickers has gained a popularity. So that also might be a reason why it was in view for more people and as than we expected. But we also need to take into consideration that out of the 16 participants, only 6 found the vegan shelf at all. So it's not a very highly representative data, but in the mean value, there's already a quite big difference. But due to the low sample size, we cannot call it significant. So it would definitely be interesting to see this done with a larger sample size. The second task's result was that the laundry detergent with simple bold lettering was fixated on the longest. But these 2 products were the most in view. This was the one that we tested mostly. And you can see that it was fixated on longer. Again, the result cannot be called significant because of the low sample size, but it can already be indicative of a difference that could be explored in a future larger study. Now, the suggestions include adding the infrared lighting to the glasses because most of the lost data was due to the low lighting conditions, and by implementing infrared lighting, the accuracy could be significantly more stable. The second is to diversify samples. So far, a very homologous type of participants have been tested. So by adding different eye color, eye shape, skin color, we could diversify the samples and train the glasses to be able to recognize data from those participants as well. To include a larger sample size, of course, 16 participants will not result in a significant data, but that was not the real goal of the research. And finally, to simplify the post-processing a little bit. All in all, the pilot study was successful. We found no significant differences, but there were the indicative ones that could be used in a larger study. And the biggest suggestion is to retest the study based on the suggestions, mostly with a larger sample size. Thank you for your attention.
Thank you so much, Luca. Indeed, was a really interesting research, and I can wait to see, what will be the next, the new updated tool. Then last but not least, we have Leonardo Menghi from the University of Trent and University of Southern Denmark that is going to tell us more about food neophobia associated with scarce olfactory performances and specific signature on oral microbiome. Leonardo, the floor is yours.
Okay, thank you so much, Maddalena, for your kind introduction. Okay, can you see my screen?
Yes.
Okay, so thank you so much again for your kind introduction. It's a great pleasure to me to be here, to be invited to this great webinar. And today I would like to show you some results, a glimpse of a study about food neophobia, and especially we measured the suitability of a paradigm concerning how food neophobia could affect our olfactory performances and host-related non-genetic factors such as the oral microbiota. So as you probably know, by food neophobia, we are referring to that trait underlying people not willing to try some other foods, and that sits at the core of the so-called omnivore's dilemma. We are used to consider food neophobia as a trait restricted to childhood, but food neophobia is still pretty common into adulthood, and this condition may lead to negative health outcomes. For example, food neophobics are more prone to avoid repeated exposures towards all that food, eliciting that sensations whose liking is not a night, like bitterness, astringency, or sourness, and this mainly leads to a replacement with foods which are more energy dense. So the results of this condition is a poor dietary variety and quality, especially in terms of fruits and vegetables, and sometimes in higher body weight. So the literature is still debating whether this condition could be or not mediated by superior chemosensory functioning or superior chemosensory responsiveness towards those sensations. Some large-scale studies, which, however, has suggested that neophobics have not superior chemosensory functioning, but these differences in the sensory responses are more mediated by an increased arousal towards foods rather than sensory responsiveness. So, unfortunately, much less is known about olfaction, and mostly host-related non-genetic factors such as the oral microbiota, which is known to be highly affected by the quality and the variety of the diets. And since the debate is still open, this contribution, so the contribution I will be talking to you in the next few slides, had aim to access the associations between food neophobia, olfaction, and the oral microbiota. So our study was attended by a gender balanced cohort of 83 FV participants, aged between 22 and 68 years old. So just imagine our protocol to be divided into two main parts. The first one was an online questionnaire through which we collected data about funiophobia and trait anxiety through the funiophobia scale and the trait subscale of the state trait questionnaire. And once participants completed the first part of the study, we asked them to reach our lab. So prior to the sensory evaluation, participants gave us a saliva sample that we later profiled by the 16S RNA gene sequencing, and then the sensory evaluations started. So at first, participants were assessed for olfactory functioning through the well-known sniffing sticks battery. And thanks to the felt pens dispensing odors provided by the kits, we were able to access the threshold and then the ability to discriminate and identify odors, so which gave us 3 different scores. The sum of these scores gave us the TDI composite score, which is indicative of the globally olfactory functioning of participants. So once the olfactory assessments was concluded, participants waited for 15 minutes and then were assessed for the retronasal aroma release of 7 different aroma compounds plus acetone as a monitor of the breath of participants during the consumption of a strawberry jelly candy. This analysis is called no-space analysis and allows us to extract for each compound several time intensity dependent parameters, such as the area under the curve, for example, and 2 different behavior parameters, such as the breathing rate and the difference in time between the time when participants put the sample in mouth and then the time when they swallow it. And we name it as duration of the oral processing. So moving to the results, starting from the olfactory assessments, as you can see from this graph, the iniophobics group, we group participants as a function of their funiophobia level, according to the cutoff proposed for the Italian population in low, medium, and high neophobics. And as you can see from this graph, the main differences was found for the discrimination and for the global olfactory functioning. So for the discrimination part, we see iniophobics that have lower scores compared to low and medium neophobics. And for the TDI, we see the same trend. It was very interesting to see also that in the threshold, okay, we see a trend through which medium neophobics had higher sensitiveness towards odors compared to low neophobics. But this difference was not found between low and high neophobics. So since no clear separation between the 3 levels of funiophobia has been found, we conclude that those differences were more likely mediated by a higher arousal towards odors. And this information was also confirmed by a trend through which high neophobics traits were more associated with trait anxiety. So moving to the no-space task, we can see from this graph that iniophobics showed overall a lower extent of the retronasal aroma release compared to the low and medium neophobics. And we see the largest differences between the groups in the so-called time-independent parameters, such as the area under the curve or the maximum intensity exhaled during the consumption of the candy. Okay. We don't see clear trends for time-dependent parameters, such as the slope of each compound between the 3 groups. So at this point, the main question is why we found those differences. And a possible explanation could be related to the 2 behavioral parameters that we extracted from the task. So the duration of our processing and the breathing rate. As you can see from these rain cloud plots, the iniophobics groups tend to have a lower duration of the oral processing, and at the same time they breathe faster during the task. So those information are quite consistent with the literature that on one end the lower is the duration of the oral processing, the lower is the extent of the retronasal aroma release, and at the same time, a higher breathing rate is commonly related to a higher anxiety. So breath can be a suitable parameter to assess anxiety from a physiological point of view. So last but not least, we also have a look to the differences between the groups for the oral microbiota. And as you can see from this correlation plot, food neophobics overall connected with several dysbiosis-related taxa microbes, okay? For example, in this slide, you can see the example of Klebsiella. So it's a genus, okay? And at species level, we can see other differences between the groups. So the ionophobic groups tend to have a higher abundance of Scardovia vixe and Klebsiella pneumoniae, which are to dysbiosis-related species, and they have a cardiogenic activity. So just to conclude my presentation, we saw with this study that highest neophobic traits showed a lower global olfactory performances overall. And it was interesting to see that higher olfactory sensitiveness in medium neophobics compared to low neophobics. And we speculated that this difference was more likely to be mediated by higher anxiety preparedness. And from a retronasal point of view, we saw that high neophobics showed a lower extent of retronasal aroma release. And this effect, it seems to be more likely to be driven by the different way neophobics faced the oral sensory contact with the sample chosen for this experiment. So taken collectively, these studies suggest a pervasive role of arousal in mediating the sensory responses of neophobics towards foods in general, which will promote a cascade system that leads to unhealthy dietary habits, and this could negatively shaping the oral microbiota. So we are sitting at the table of the future. So which could be the future of this research? And as you probably saw in the correlation plot, there were lots of correlations between the threshold. So the more physiological part of the olfactory assessment and several genes in the oral microbiota. So the main question we could answer in the next future is whether our sensory responsiveness could be or not affected by the oral microbiota. So my presentation is over. I would like to thank you so much for your time. And if you have any further questions, please feel free to ask.
Thank you, Leonardo. That was a fascinating talk, and definitely I think it's great that your project is trying to combine many different disciplines. I think you're a pioneer in doing all this, working with microbiota and relating to other aspects. So great. There are some questions in the chat. So we will start with the second and final discussion table. The first questions are addressed to Isabel. So thank you for your nice presentation. How did you choose the TDA ballot? And were the consumers habitual consumers of ice cream and pizza?
Thank you for your question. Regarding the consumers, yes, we recruit participants who consume ice cream and pizza at least once a week. So we are participants familiar with the product. Yes. There was another question. Could you please help me, Karina?
Yes. How did you choose the TDE ballot?
Because you mean the technique, the band plots? Why we choose that one? Is that the question?
I think maybe, yeah. How did you choose the emotion that you asked during your
assessment?
Yeah. So there were specific emotion. Did you consider also maybe other emotion for this?
Thank you, Madalena. Yeah, regarding the emotions, the emotions were based in a pilot from a previous study. So for the pilot, we selected 32 emotions from literature review. We did a pilot with 20 participants to select the most frequent, and we selected the most frequent terms that they chose for those products. And then we did another pilot, with those selected emotions, were about 20 emotions. And then the 10 emotions that are in this study, were a combination from the selected terms from the pilot plus the available emotions from the software face reader that we use for the facial recognition. So the combination and the terms, the most selected terms that match both criteria, we use in the study.
That sounds really interesting. It might be interesting also for the future to consider more, I would say, all complicated emotion, but I guess that's of course requires also from a lot of validation first, so a lot of more pilot studies. So congrats again with your research. We have then a couple of questions for Luca. Can you please explain us a little bit the difference of the parameter that you analyze? So the in-fixation one and the in-view one.
Yes, of course. So the difference between the two values is that the in-view value takes into consideration when the object is in the visual field, but is not directly looked at, while the fixation only measures when it is looked at, but not when it's just generally in view.
Oh, okay, really clear. And yeah, we had that. So another question: can you think? Well, you tested in a supermarket, but can you think perhaps other settings that where this tool might be useful?
Yes, of course. In terms of measuring products, I think supermarket is the best choice. However, if we do not necessarily stick to the wearable eyeglasses, or even if we do, looking at web shops and online shopping could also be a very, very good alternative, also because it's more controlled environment, so it will be more reliable in terms of data. Yeah, but if we're looking at very, very specific label designs, then a completely controlled environment can be suitable as well.
Oh, thank you so much.
Yeah, definitely this is a tool for the future, tracking where your eye is looking at. Now we have questions for Leonardo. Have you ever evaluated expectation dealing with neophobia? How did you do it? And compliments on your presentation.
Oh, thank you so much. Thank you so much. Actually, no, I haven't evaluated expectation on food neophobics. But I would expect that food neophobics tend to react in a more involved way compared to neophilics person. So they have more negative expectations toward food. So because they just think about the experience that they are about to live. So especially when those foods are more characterized by that sensations that in my presentation was defined as not a night, like the bitterness, the sourness, and astringency, and so on. So maybe it could be a matter of attention in this case. So they are more attentive towards those kind of foods, and so the avoidance results is more clear compared to other group of people.
Thank you. And in your opinion, how could the oral microbiota affect the perception of tastes and of smells?
This is a very fascinating topic, and actually it's the main topic of my PhD, so that's the reason why it's fascinating. No, I'm joking, of course. Actually, there are several theories behind this putative association between microbiome in general and taste responsiveness. So the first one that is quite accepted by the community is that oral microbiota could use tastings for metabolic purposes. So it's a matter that tastings will not reach the taste receptor that we have in our tongue, and this could affect the way we perceive from an intensity point of view the taste from the food. Another theory is related to the formation of biofilm. So the biofilm, actually the result will be the same. So there are several biofilm producers microbes, for example, the ones that I was presenting before, and that could actually put a layer on our tongue, and this could avoid testings to reach the receptor, and then limiting all the jurisdiction of the signal, so of the process.
Yeah, definitely. I think it's fascinating what you're doing. So great, good job, and good luck with the rest of your PhD.
Thanks a lot.
We have a question for all our speakers, so feel free to open your mic and answer to this question. So where do you think future issue in healthy eating will be, and why? So take your time.
I might jump in because I think this is a good question. From my research perspective, if people are shifting their consumption habits away from meat, I think it's quite valid to think about what they're substituting it with, whether it be fish or plant-based alternatives, and how healthy they are. So I think, yeah, food preferences for these alternatives might be a challenge for the future, and whether they're nutritionally balanced.
Yeah, indeed. Yeah, I think this is also a topic that will a lot of investigate by Hannah Giuliani with their children. Maybe they will be the answer for this. Is there anybody else that would like to say their things about this question? Then
okay, then with that we would like to conclude with this discussion table. So thank you all for joining. Thank you to our great audience and for being active in the chat with your questions, and for staying with us for this good almost 2 hours. So you audience deserve a nice clap. But especially, we want to thank our presenters of today for sharing a little bit of their work. And I really think that they are doing very fantastic and very relevant research. So you definitely deserve a very big clap.
Before we leave, yeah, Nicolas just mentioned before the webinar, we want to remind you that in just a month from today, we're going to meet up again in the 10th anniversary, at the 10th E3S annual symposium. So if you haven't registered yet, we're going to share with you the link where you can register and read a little bit more of the program that will be there. So we will be there. So if you are also interested more on knowing what we do or being part of this association, please, you can find it there. Last but not least, yeah, if you liked this webinar and you want also to be part of this organization, we are looking to expanding our board. So right now we have 2 available positions. So if you're interested to know more about this, please send us an email, or you can also visit our page in the E3S website. So with this last slide, we thank you everyone for participating, and yeah, we wish you a lovely evening or morning, whatever you are.
So thank you and bye.
Bye.
Thank you. Bye.
Bye.
Hi, Jonah. Thank you so much for setting everything.
Yeah.
Oh, I can't hear you.
Yeah, you're muted.